Hello, this is Jesse. Today I'm going to be going over vector design. The objective of this tutorial is to design an expression vector in silico that will be used to express a protein of interest in E. coli. In this example, our protein of interest is myricin 1, an alpha helical antimicrobial peptide. We will be using Genome Compiler to design our expression vector and Snapgene to retrieve Plasmid file to work with. Below is the sequence for myricin 1. And then here's an overview. So our goal is to take a gene, insert it into our expression vector, and then this will be done in silico. So to test whether we're going to be planning this out to see whether once we take the gene of interest and put it into a plasmid, our expression vector, if we were to insert it into an organism such as E. coli, will our organism express our protein correctly? Is the reading frames correct? Do we have it in the correct direction of the promoter? Okay, so we're going to be trying to plan and verify that we have our gene of interest in the correct place. So we're going to be going through this step by step. So first, I give y'all a group of URLs to use. So first, we're going to be going over gene design for reverse translation. So what we need to do is we can take our protein of interest and open up gene design. I already gave you the URL right here, so just copy and paste that into your browser. And we're going to reverse translate this protein sequence into a DNA sequence. So here we have protein, and we're going to convert it into a DNA sequence. We're going to codon optimize our protein sequence for E. coli, okay? So if you notice, if you click here, some of the codons will change. Well, this is because E. coli appears to have more of a particular type of codon compared to other organisms. Remember how some codons can code for the same amino acid, and some codons will appear more often in certain organisms compared to others. So E. coli has a preferred set of codons. So we're going to codon optimize this. We're going to codon optimize the resulting DNA sequence for E. coli. So we click reverse translate, and then here we have a DNA sequence that has been codon optimized for E. coli. So what we will do is we will copy and paste the sequence and we can save it. Here I already have it done for you. Here, codon optimize DNA sequence. Next, we're going to grab a plasmid. So on the top, we're going to need a plasmid, right, where our gene of interest is going to be inserted into. So here we can go to Snapgene, which is a website and a service that can be used that holds plasmids and also has some softwares to visualize plasmids. And we will use Snapgene Viewer to visualize this plasmid here. So here I already gave y'all a URL to this plasmid file, PET28. PET28 are a group of plasmids that are used for bacterial expression. PET vectors are used for bacterial expression. A group of them are. And in this case, we're going to be using specifically PET28A. Okay. And I gave you the URL to PET28A, and you can download the PET28 file here, and we can visualize it with Snapgene Viewer. So you can just go here to download Snapgene Viewer. It's a free software, and we can open up Snapgene Viewer. once we open up Snagene Viewer, we can open up our downloaded PET28 file. So we can click on Open, Open Files, and then we can take find where you saved your file at, your PET28 file. I already have mine saved here, so I'm going to go here and open it up, PET28A, and then we can open up the file. We can go to View and View Map, and then here is the Plasmid map, okay? And the software even annotates the features of the plasmid. So we have our multiple cloning site where all these restriction enzyme sites lay. Then we have our T7 promoter, which is going to go on the bottom strand, right here, the bottom strand. And it's coding anti-clockwise here, okay? And then we have some restriction enzyme sites. We can choose either of these restriction enzyme sites, okay? But to get an idea of what restriction enzyme sites we want to use, we need to figure out what restriction enzyme sites are in our sequence here, our DNA sequence that we just got from codon optimizing it. So I'm going to copy and paste this, and I'm going to open a website called nebcutter, and I'm going to paste my DNA sequence. Remember, this DNA sequence, if you translate this into a protein sequence, you're going to get the same sequence for myosin up here. You're going to get the same protein sequence, okay? But we're going to verify and check and see whether what restriction enzyme sites are located here. So here, if you look at our PET plasmid, I want to pick EcoRI and BAMHI. These are two that I want to pick. I can choose any of these if I want to, or if you want to, you can choose any of these, but I'm just going for EcoRI and BAMHI. Okay. If I go to nebcutter, notice how if I look through every single one of these restriction enzyme sites, there's no EcoRI or BAMHI, right? None of this here. So I can go ahead and choose BAMHI and EcoRI. Because if I were going to do this experimentally, and if I were to have my gene of interest, and I were to use enzymes to cut my gene of interest out from another plasmid, it would not cut my gene of interest anywhere where I don't want to be cut, right? I don't want from 1 to 198, I don't want my gene of interest to be cut anywhere in here. But on the outside, where the restriction enzyme site should be, that's where I would want it to cut only. I'm just double checking here to make sure it's not going to cut anywhere. And it looks like I'm good to go with my restriction enzyme sites BAMHI and EcoRI. So this case, notice these arrows are pointing in certain directions, okay? These arrows indicate where the reading frame, where it's being read. Okay, so we have T7 promoter pointing counterclockwise, okay? This direction, counterclockwise. That means the bottom sequence is going to be coded for. So here we have the 5' in, 3' in. So this is the top part right here, and then we have 3' in, 5' in. Our DNA will be coded, will be reading the bottom strand right here. The T7 promoter will be reading this bottom strand because it's pointing counterclockwise, right? So that means we need to insert BAMHI will be on the 5' end. BAMHI will be on the 5' end. And then EcoRI will be on the 3' end. Okay? Okay, so we're going to take these EcoRI is going to be on 3' end, BAM is going to be on the 5' end. Okay, and our DNA is going to be coded from 5 to 3' on the bottom strand. Okay, not the top strand. If, say, it was pointing in the same direction as this LAC-I promoter and LAC-I repressor, then we would be focusing on the top strand. We would focus on top strand to be coded in clockwise direction. I hope that makes sense. So we're going to go down and we're going to continue on. We're going to download our file. Well, we already have it downloaded and we already picked where our restriction enzyme sites are going to go. BAMH is going to be a 5' end, EcoRI is going to be on the 3' end. And then we're going to use Snap genome compiler to actually do the in silico construction. We're going to use it to double check whether the reading frames will be correct and whether we already have it planned out in our head. We just need to double check to see everything's going to work out properly. So we can take new construction, click on new construction in genome compiler. Before I get ahead, just go to genomecompiler.com and then you can open up, or you can create an account. Let's go to sign up, it's free, and then I already have an account. Once you have this set up, we're going to go to restriction ligation. Just remember, click on new construction, in-house cloning, project name. You can name whatever you want it to name. I'm going to continue on and I'm going to click restriction ligation. I'm going to go to next. Then I'm going to click, you can take your file and you can drag and drop, or you can search for it. I'm just going to click on choose a file, and I'm going to go to my documents, and I'm going to go to where I already have the PET28 plasmid saved. So here, plasmids, PET, and I'm going to go to PET28. So my backbone is going to be my vector, my plasmid, and then my insert will be the gene of interest that I want. In this case, it's going to be myosin. So once this loads, I'm going to click on digest. I'm going to digest with EcoRI and BAM-HI. So I go to BAM, BAM-HI, EcoRI, EcoRI. I'm going to find EcoRI. Okay, then I'm going to select the backbone fragment. I'm not going to click the 2-pair base length. I'm going to click the 5359 base pair length. And so here we have BAM-HI. Okay, BAM-HI right here, and then EcoRI right here. Okay, we're focusing on this bottom strand, G. So from 5' to 3' will be the C right here. So this bottom strand is what we're focusing on. Next, we're going to click our insert, click the insert button over here, and we're going to paste in our DNA sequence that we got from reverse transcribing the myosin protein sequence that we got in the very beginning of this tutorial. So we're going to copy, paste here. I'm going to use this as an insert, linear, and we're going to do linearized by synthesis. Okay, and we're going to add our restriction sites. We're going to go to BAM, and we're going to BAM B. Okay, and then we're going to BAM-HI for a 5' end. Remember, earlier we already planned out that we want the 5' the BAM-HI to be on the 5' end, so it's going to go in the correct direction, the arrow, the reading frame is going to point in the correct direction on the plasmid map. EcoRI for 3' end. So we'll scroll down to find EcoRI. Here it is. And it's giving us an error. Well, we can fix that. Strand forward. So right now we have BAM-HI and EcoRI. Or you can say that our gene of interest is pointing 5 to 3 for the top strand. We're going to reverse it, fix it, and now when we finish right here, when we click next, here's EcoRI, product, BAM-HI. Next, you're going to see that our product is pointing in the direction that we want, in the same direction of the T7 promoter, okay? And from here, we're going to we're almost done at this point, okay? Notice how, if we look at the map here, sometimes it might be a little bit hard to find the T7 promoter. Here it is, T7 promoter. This is where the T7 RNA polymerase, a certain polymerase will bind to, and then it will code along right here, and it will code for our DNA right here, and create an mRNA that will eventually be translated into our protein of interest, myosin. Okay? Supposedly we'll have our correct sequence, but we need to check by using open reading frames. Okay? So you click here, making sure everything's good. You can click finish. Yes, we can name it. I already have this named. I already have a sequence name, but I will create another one. Example 1000 and click OK. Oh, downloads. OK. No. And we can click on the file that we just saved, or the plasmid we just saved with our gene of interest. Okay. This is what we're interested in right here. Right here. So here it is. It's already highlighted for us in the orange. But what we can do is we can click View, and we can click the Open Reading Frames. Click Open Reading Frames, and then it will give us the Open Reading Frame, and we have here our sequence, our DNA sequences are here, and then we have our protein sequences, the corresponding protein sequences above. Okay, and what we're going to do is we're going to find where our protein starts, and then we're going to find where it ends, and then see if we have the correct reading frames. Will our protein of interest be coded for? Okay, so here's our first sequence. This is where the insert is. You see BAM, and then we have second frame, frame 2. We have frame 2 right here, M, N, I, K. So we have methionine, spargenine, isoleucine, and lysine. So we can double check. Going to the very top. Methionine, spargenine, isoleucine. So that was the second frame. And then the last bit we had right here is our ending sequence. Arginine, lysine, and histidine. Arginine, lysine, and histidine. So it's safe to say that we have our gene of interest. Our gene of interest is in the correct reading frame, because when we look through the reading frames, the second reading frame indicates that we will get this protein. This protein is a resulting product. And if you don't like using Snap Gene Viewer to verify that you will have your protein of interest coded for, then you can use Snap Gene Viewer. So we can go to Snap Gene Viewer, and we can upload our saved file, our saved plasmid that we just created, but we need to export it first. So we can go to example 1000 or whatever you named it, and we can click export. I'm going to export it as a FASTA file. I'll put it to desktop, and then I'm going to save it. I already have one saved. It's the exact same thing. Okay, I already have it saved, online example.fasta. So after you're done exporting it, you can go to open, and then you just open up your file that you just recently saved. So I'm going to desktop and go to where you saved your and I'm going to make sure format options for this file, I'm going to make sure circular is selected and automatically annotate common features. This will show me the features that are in my plasmid. It's going to show me the protein, the lac-I promoter, the repressor, cloning site, the antibiotic, origin of replication, and all that stuff. Okay, so make sure you click annotate common features. Then once it opens, we have our gene event. Now we have our plasmid, but notice how we have this gap. Well, the cloning site's not here anymore, which is good indication that we have our protein, our DNA sequence coded right here. Remember, we have BAM-HI. We inserted our gene in between BAM-HI and EcoRI, and here that is. And then we have our T7 promoter. Now, I'm just a fan of the Snap Gene Viewer. It's easier to find the sites. Okay, I prefer using BAM Snap Gene Viewer when I'm verifying whether I have the correct open reading frames and whether my protein will be encoded correctly. When I actually take this gene, or take this plasmid, this expression vector, and I transform it into E. coli, and I induce the E. coli to express my protein of interest, I will get my protein product. Okay, and I will go to view, and I will go to sequence, and then here we can find BAM-HI right here. Okay, remember we had our first three first three amino acids are methionine, asparagine, and isoleucine, and our last three we look for EcoRI right here. So we have histidine, lysine, arginine, and you can double check and verify that yourselves. This histidine, lysine, arginine. Histidine, lysine, arginine. And you can go all the way back if you really want to. You can go through every single amino acid, and I guarantee that in between EcoRI and BAM-HI will be our protein of interest. And we will have some additional sequences, right? We have some additional amino acids here. But if you really wanted to, you could add if you just say I'm like, okay, look, I have my protein of interest here. I have the tag here. I even have the thrombin site. So thrombin site to cut. I even have a histidine tag here in my protein. So I'm gonna be able to purify my protein. I'm going to be able to remove the histidine tag with the thrombin site. And then I have a T7 tag, and it even tells me what it's for. It's for efficient translation in E. coli. But at the very end of my protein, in this reading frame, after EcoRI, after this part, I have some extra amino acids here. Now, this might be okay, just depends on what you're trying to do. In a homework question, this is fine. I will accept this, I will take it. But if you really wanted to stop, if you did not want these extra amino acids here, then you would have to either change frames or add a stop codon here. Okay, you can add a stop codon after histidine. Just add a codon in between histidine and glutamine. Not glutamine, glutamic acid. And then from here, you're going to be able to insert the stop codon here, okay? And you're going to be able to prevent these additional amino acids here, okay? And from here, you'll be fine. You won't have to worry about these additional amino acids, okay? And the thing is that why that's possible is that now you can have your DNA sequences synthesized. So if I go to company, GenScript, gene synthesis, this company will synthesize your genes for you. Okay, you might have a gene of interest synthesized, and you will need to give them your protein, you will need to give them your gene sequence. You just literally give them, if I wanted them to synthesize a gene that would express, or not a gene, but a DNA sequence that would be translated into this sequence, this protein sequence, I would give them this site right here. I would, while I'm going through the process of constructing and making my order, I would give them this DNA sequence right here, okay? And say at the 5' end, right here, this 5' end, and then 3' end. 3' end is where I would need to add the AUG here. And then that would be a stop codon, correct? And this would be able to, this would, or not AUG, but I would put a stop codon here. You can look up the stop codons and choose which one you want, or whatever one is actually optimal for E. coli. And from there, I would have to put UAG or UAG, and then this would be T. So this would be the stop codon here. Add that, and then give, and this right here would produce a stop codon here, and it would prevent these amino acids coded for. But notice how I can change it, and the company would give me this gene sequence, this DNA sequence, and a plasmid. When you're choosing the plasmids, it could be in a cloning vector that doesn't have a T7 promoter, right? And then you would need to cut out your gene of interest. So on the very top here, cut out your gene of interest, insert it into your expression vector, which we just did. We went through the whole process of taking a gene of interest, right here, we went to this protein sequence, and we inserted into PET28 vector. And then at the very end, we were able to verify that the reading frames were correct. We will have our protein sequence being encoded. The protein sequence myosin 1 is going to be encoded in this plasmid. Okay, and then in the next tutorial, I will give you a case where we will be expressing, we will insert 2 proteins of interest into the same expression vector. Okay, so this is the end.